screen?
I can see it.
Okay. Okay, so our project was on the evolution of multi-step traits in different social structures.
Yep. And so both Roshan and I have always loved animals and nature, and we really wanted to investigate this passion from a more quantitative perspective. We were fascinated by the diversity of animals in our environment and naturally turned to evolution to understand how these complex traits have evolved. So we wanted to come up with a really accurate way to simulate evolution because we felt as if that would be a really great thing to do within our project. We were particularly interested by how behaviors have evolved and how animals can live in various different types of hierarchies and how these different social structures can affect the development of many different complex multi-generational traits within their populations. So in here, we have our awesome example. Our favorite is the Eclectus Parrot. And so the Eclectus Parrot represents one of our social structures which we did. So the male Eclectus is in the middle with all the female Eclectuses on the side. And the way what this represents is that the male Eclectus is the central node of the population, with all the female Eclectuses being on the side. So the male Eclectus goes and individually breeds with each of the female Eclectuses within the population. So that single male Eclectus, since it's the most dominant within the forest, is going to be spreading its genes pretty much throughout the entire population, with rarities where other male Eclectuses are able to come in. But for this experiment, we just decided to have it be a perfect population. And as you can see, there are many different social structures. We decided to go ahead and simulate 3, where these different social structures were the homogeneous, meaning that all of the individuals were interbreeding throughout each other, kind of like rats or something. The star, as I just explained, with the Eclectus, and then the superstar, which is a lot more of a complex social structure, where one individual is going off and breeding with different communities of individuals. So, and as I said before, but I didn't necessarily state it as the hypothesis, our hypothesis was that these different social structures affect the development of these multi-generational traits. So, what we did was we tested this hypothesis and we used something called the Moran process with our hypothesis. So the Moran process is a very respected kind of way of going about things within the biological community, introduced by Patrick Moran, and it's a stochastic model for evolution within a population. And basically, within the Moran process, each individual is given a fitness that determines their likelihood to reproduce, and at each, and so the Moran process goes over many generations, and at each time step, one individual gets to reproduce based on the fitness, how fit they are, while another individual is chosen for death. And then the individual which is able to reproduce has their baby, the then occupying the position of the dying one. And basically, so this is repeated until some species takes over the entire population, and the probability that this occurs finitely over many iterations is 1. This is an interesting and counterintuitive fact that although it is possible for multiple species to coexist forever, in this model, it occurs with the probability of 0. So within our model, we had a few assumptions that helped us kind of make our model a bit more accurate. So our assumptions was that it had a fixed population size, meaning that it never fluctuated. The number of individuals never fluctuated. And every species was pretty much determined by a fitness rather than a bunch of genes combined. We had just the fitness of a single gene to determine the population. And the more fit individuals are more likely to reproduce. And since the population is homogeneous, where some where they are chosen at death for random by the Moran process.
Okay, and one thing I'll add is just that this assumption is sort of what other models that we use later on challenge. And so a bit on the notation and terminology that we'll be using for the rest of our presentation. One is fixation probability. And this is the probability that a beneficial mutation will eventually take over the entire population after some finite number of iterations in the Moran process. And this is denoted as rho. And as Jacob mentioned earlier, the probability that this doesn't occur is 1 minus rho. So either the population fixates or it's called drifts, in which case the mutation goes extinct. Another important concept that we've been using in this project is the idea of graphs, which are mathematical representations of a network. And this can represent really anything, but in our case, it's representing individuals and their evolutionary interactions. And sort of algebraically, the way a graph is described is with an adjacency matrix. And this is really has entries containing values for each of the strengths of the edges. And I'll go more into that in a later slide. And finally, the Moran process is a Markov process, which means that the likelihoods of transitioning to a different state at each step depend only on the current state that the system is at. And so one of the benefits of this is that people have been able to use this to make statements about the Moran process and also its extension to non-homogeneous populations. And so, yeah, that extension is sort of what we based our project off of, and a paper from 2006 called Evolutionary Dynamics on Graphs. And what they do is they make an alteration in the Moran process that at each time step, rather than choosing an individual for death completely at random, an individual is chosen for death according to its relationship within the social network. And this is where graphs come in, where the outgoing edges correspond to the probability of an individual replacing its neighbor. And this can be illustrated in the image in the bottom right. For example, individual V3, if it's chosen to reproduce, the probability that it replaces V1 or V5 is given by W31 and W35 respectively. And mathematically, that exists in the third row of this matrix, where you can see W31 is entered and W35 is entered, whereas the rest of the entries are 0 because it's not able to kill off those individuals. And so what are sort of the key findings of that paper and that sort of, I guess, inspired our project? Well, sort of most importantly is that certain structures greatly improve the likelihood of a beneficial mutation taking over the entire population. And so another way to put that is that for at least single-step mutations, the social structure has an impact. And so our approach to this and our model was to look at this similar algorithm, but try to extend it to what if we're not just looking at a single change, but a change that occurs over many generations. And this is the motivation behind this is that many traits that we see in nature don't occur because of a single mutation. They occur because of a series of mutations that occur over many steps. And so one example of that could be like a wing, where, you know, it doesn't go from one generation not having a wing to another generation having a wing. It builds up over many generations. And so we simulated this in 3 different social structures using a Python program. And we'll go into a bit more in depth on those specific structures in the next slide. And we simulated the development of a trait which takes many generations to occur. And so the way that we sort of introduce this into the model is we assume that at each time an individual reproduces, there is a certain probability that its offspring will exhibit a mutation in the next step in that sequence. And so in which case it would have a higher fitness. And so a bit on the specifics of what we tested, we used 31 nodes, which in each population structure, which may seem like an arbitrary number. And the reason we did that was because of one of the structures called the superstar has these very specific parameters that lead to having these sort of strange round numbers. And so here is an illustration of the models that we are of the social structures that we tested. First is the homogeneous, which you can see is where each individual is interacting with every other individual without any specific pattern behind that interaction. Secondly, you can see the star, which is really there's a central node that then interacts with these other nodes that are not necessarily interacting with one another. And then finally, we looked at the superstar, which is where we have these sort of subgroups in these hierarchies. And so the key result here, and these formulas were derived in that paper, is that fixation probability, or the likelihood that a mutation will take over, is actually amplified depending on the graph. And so you can see for the first one, the probability that is 1 minus 1 over r, whereas for the star, it's 1 minus 1 over r squared. And so if r is a fitness, squaring it, since it's greater than 0, will naturally make it larger. And so then dividing by that will create a smaller number, and then subtracting that will make it closer to 1. And so in the superstar, actually, and this is sort of remarkable, is that depending on how many leaves and how many leaves, the amplification parameter can actually be to some arbitrary power. And so we're really raising it to however large we want based on how we organize the structure.
Yes. And so here, as you can see, this is part of what Roshan was talking about a little while ago, where the fitness was changed each generation according to the different rules that we had set. So within our simulation, we decided to simulate it in 3 different ways where the fitness changed. So here, the fitness changes linearly, and at different stages of the mutation. So there are 5 different generations of stages where the fitness changes, and that was one of the unique things about our project, which we changed up from the paper in Nature, because we wanted to show that we were doing unique, cool science rather than just copying something from Nature, and we wanted to see how the fitness really changes the way the Moran process works. So as you can see here, the fitness goes up linearly, exponentially, and oscillatory. Oscillatory is where it starts up, goes down, starts up, goes down, and the other are pretty self-explanatory. And in each of these, there are 5 different stages of mutation where the fitness differs.
And in terms of our experiment, the linear graph can kind of be seen as our control, whereas the exponential and oscillatory ones are sort of where we're changing it and testing something new. And so here are our main results from doing that simulation. And firstly, our sort of overarching hypothesis that we mentioned at the beginning of the presentation was supported by this, because in each case, the superstar and the star were tested to be significantly different from the homogeneous structure. But some interesting findings here. And so I guess first I'll discuss what's expected. What is expected was that the star would be higher than the homogeneous structure in each case. But something that was unexpected was sort of how poorly the superstar performed in our simulation. And one reason why this could be is because of the fact that we're limited by the number of iterations that we can go through on our code, because the superstar is so complex that the runtime grows very quickly, and because the superstar is a very heterogeneous structure in comparison to the star or the regular Moran process, it sort of lends itself to there being certain nodes that change very rarely. And this is why it requires a much larger sample size to sort of see a more accurate probability. And then another interesting finding is that the linear versus the exponential curves didn't actually make a huge impact on fixation probability. And this is surprising because if we go back to this slide, the exponential fitness goes to a much higher absolute value. So for example, at the final stage, it's about at 16, whereas the fitness in a linear curve only goes to about 5. Yet the fixation frequencies that we simulated were quite close for both of those. And our reasoning for that is that we think that maybe, it depends more on the shape of the curve or the direction that it's going rather than the actual absolute difference that it's reaching. And this is kind of ties into the fact that this model is very abstract. Even though it's representing things in nature, it's representing them in a sort of mathematical way. So for example, fitness is for us, an arbitrary measure of how successful an organism is. But the task of looking at an organism in an environment and saying, this has a fitness of 5, that would be a completely different task and would be really difficult. But yeah, so and another issue that this presents is that maybe the reason why the exponential and the linear are fairly close is that the exponential curve actually means that fixation occurs much faster, but still at a similar ultimate frequency, and that would be something that we would have to test later because we didn't look at the runtime of our simulation.
Yes. So as you can see, the stage of fixation. So in our model, fixation only occurred at the final stage and never at the intermediate stage. So what that means is that on those graphs before, where it said linear, exponential, oscillatory, they had 5 different stages of mutation. So the only places where the fixation occurred was on the final 5th stage, never in the intermediate stage. And that makes sense because the individual progression up the stages are single-step evolution instead of a multi-step evolution. So it makes sense that the final stage is where the fixation occurs. So that would mean that runtime, how long we run the evolution for, would be very influential, and that is not a variable investigated here, which could be important for future work. And this means that fixation may hinge on whether or not a deficient fitness lowering intermediate mutation is able to survive for long enough to produce an offspring in the next stage with greater fitness. So that's something which we could potentially look into in the future, is how the runtime affects the fixation, since it never occurs at an intermediate stage and is always occurring at that 5th mutation sequence. And so so then in our conclusions, so we developed an algorithm in Python and we computationally reproduced the results from the paper in Nature, which we based all of our entire project off of. Of course, we changed it up a little bit with the mutation stages, and we we made more complex multi-step traits, and we modified the existing model of the Moran process to do this. So, and then we analyzed the biological significance of these traits. We also did really well in the Denver Science Fair and won the Mu Alpha Theta Award and moved on to the Colorado Science Fair, which we thought was really cool. So some things which we could do in the future is we thought it would be really nice to work with the computer scientists or run it on a supercomputer to see how the code to see if we can run things at much larger numbers, because that was a big thing for us. Was it was taking such a long time to run the larger numbers that we couldn't do it, so we had to deal with smaller population sizes, and we could try to find a way for our model to work with the real world and show these results to biologists. So we thought it might be really cool to talk with biologists and identify our theoretical results might be significant in nature, and we would love to use our mathematical proof to find the exact differences in fixation probability as well, because we think that would be a good further step.
And finally, we'd like to say thank you to our mentor in this project, Professor Pankovich. I think he's in the Google Meet right now. And also thank you to everyone for listening to our presentation.
Yes, thank you.
And here are our references.
All right. Well, let's first give them a round of applause. That was amazing. I love the use of the math for this. Like, I can really understand it when I started thinking about it, but the fact that you've been able to put some math to these biological terms is amazing. So first, let's open it up to the class. If you have any classmates that have questions, please go ahead and ask these two gentlemen right now.
Yeah, I had a question, or maybe a suggestion for future presentations. I am not too knowledgeable on the biology and the things you guys know, and I definitely had a little bit of trouble trying to figure out what's happening with some of your background research.